Hi, my name is Jong-Suk Park, PhD student at Georgia Tech. Our demonstration is about high-density multimodality CMOS sensor array for cell-based assay. We will first talk about the motivation of our work, and then we will show our cell-based biomeasurement result. First of all, a cell-based sensor assay is a hybrid system composed of living cells and sensor electronics. Living cells is directly used and directly cultured on the sensor chip surface, and therefore the living cells can be viewed as a biological front end. The sensor electronics measure the living cells' physiological change and then convert them into electrical signals. Therefore, the sensor electronics can be viewed as electronics backend. A cell-based sensor assay has been widely used for cell characterization and drug screening due to its inherent advantages. First of all, it directly uses living cells, so it provides physiological relevant information. And also it provides a more accurate representation of real-life model of living cells. Also, it achieves high sensitivity and detection can be very fast. Recently, cell-based assay has gaining more interest for high-throughput drug screening. For example, it has a great potential in personalized medicine, as the patient-derived cell can be directly used to test efficacy and toxicity and potency of a newly developed drug. However, cells are a highly complex system with a concurrent multi-physics and multimodality response under external stimuli. This is our motivation of work. Therefore, we propose and develop multimodality CMOS sensor array to simultaneously capture the multi-physical cellular physiological change and enables holistic cell characterization and drug screening. For example, action potential and local field potential can be measured by a voltage recording modality, and the cell growth and cell attachment can be measured by impedance mapping. Also, the cell morphology change, opacity change, and cardiac muscle contractility can be captured by optical detection modality. As a second part, we will present our cell-based measurement results.
I'm Moaz Aziz. I am a PhD student at Georgia Tech. So as my colleague mentioned, we propose a CMOS multi-modality sensing array for holistic cell characterization and drug screening applications. Here are some measurement data that we did in order to put these ideas in action. So this is a stereo microscope image of on-chip cultured rat cardiomyocytes. These are GFP-based cardiomyocytes here. This is the optical shadow image. Notice how well it correlates with the stereo microscope image. We can clearly see where the cells are located. And if we zoom into one of these groups, here is an impedance mapping of that group. Impedance mapping is very useful for cell attachment assays. And as we can see here, this impedance mapping is demonstrating that this group of rat cardiomyocytes is well attached to the CMOS chip surface. In the case of dissociated culture, strong cell attachment is a sign of solid growth for the cultured cardiomyocytes. Here is also the extracellular potential recording of this sample. As we can see here, each of these spikes represents an action potential event consisting of a depolarization and a repolarization. Furthermore, we have also demonstrated the ability to measure different aspects of cell physiology using other types of cells, such as human ovarian cancer cells. This is a HEA8F8 sample of human ovarian cancer cells. And here we are determining the morphology of cancer cells, which is obviously a very useful aspect when it comes to determining how cancers are spreading. In addition, we also did a growth assay of bacteria. So this is the initial bacterial colony. We basically took a plate of agar with bacterial colonies on it. We cut out a sample colony. We went ahead and put it on the CMOS chip, and we monitored its growth. This is the initial condition. This is 3 hours after afterwards from the incubator, and this is 1.5 days afterwards in room temperature. And you can see here the bacterial colony has grown, and then it extends beyond the sensing area, and you can see that our CMOS multimodality sensing array is successfully able to capture that. In addition, it is also very important to be able to determine how different modalities of our sensing array are related to one another, how different features of our of what we capture in our data are relevant to one another. And this plot down here illustrates that quite nicely. Here we have action potential recording of cardiomyocytes, and we also have contractility recording, i.e. optical shadow imaging, that basically represents actual contraction events that we can see under a microscope. So basically, as you can see here, we have a change in the optical shadow imaging light intensity, right, as each particular contraction event is taking place. And we can see that the frequency of contraction events is reasonably well correlated with the frequency of action potentials taking place. So that takes care of cell characterization, but another key application of our multimodality sensing array is drug screening. So here is the action potential recording of these rat cardiomyocytes before isoproterenol administration. As you can see, the beating has a medium frequency, and the amplitude is on the order of approximately 1 millivolt. However, after administration of isoproterenol, which is a cardiac drug that's designed to increase beating frequency, you can see that the frequency increases significantly, and the amplitude also drops, becomes on the order of 500 millivolts. So as you can see here, we have clearly demonstrated that our CMOS multimodality sensing array is able to extract multiple features of cell physiology. We are able to post-process these data in such a way as to be able to determine how these features are related to one another. And furthermore, we are also able to use our multimodality sensing array for drug screening applications. Case in point here, in the case of before isoproterenol administration and after isoproterenol administration, where we are able to observe a change in frequency, significant change in frequency, and a diminishing of the amplitude of the action potential. This is our fully packaged CMOS multimodality sensing array chip. We have the chip in the middle, we wire bond and seal it, and then we add a Petri dish on top in order to accommodate liquid phase media. This board here serves essentially as a daughter board, which is then connected to an associated motherboard. This motherboard-daughter board is itself self-contained in the sense that we don't need to have additional electronics from a biology lab supplemented to this setup. This entire setup itself can run by itself in a biology lab and can be connected to a computer for data acquisition. I'm Moaz Aziz from Georgia Tech. Thank you very much for watching.